Since laundry detergents have many harmful effects on waterways and living creatures, our goal was to create a more effective and eco-friendly enzyme-based process for washing clothes. In order to hydrate oleic acid and make it more water-soluble, we started by creating a recombinant plasmid with a gene that encodes for oleate hydrase from Elizabethkingia meningoseptica, and then E. coli can be used to create oleate hydratase enzymes to create the oleic acid. Degrade the oleic acid.
In contrast to detergents currently in use that harm waterways and aquatic life, enzyme washes break down as organic waste.
To produce oleate hydratase, the DNA sequence from E. meningoseptica that codes for oleate hydratase is inputted into E. coli in a recombinant plasmid. Once the oleate hydratase is produced, the enzyme will be purified and extracted.
The oleic acid is then used as the input for stain removal. Once the enzymes have acted upon it, 10-hydroxysteric acid is produced as the output.
We transform E. coli to produce the enzyme oleate hydratase, which can convert the nonpolar oleic acid into a more polar form that can bond with water more easily and be washed away. Since oleic acid is a common non-washable dirt molecule, this helps make the fabric more easy to clean in the long run.
By using both sticky and blunt end restriction enzymes, we can cut out a section of the E. coli plasmid and attach the genetic sequence coding for oleate hydratase to the plasmid. This enables us to form an operon that forms part of the pathway that creates oleate hydratase proteins.
This research will be conducted in a clean and safe lab with surfaces and equipment thoroughly sanitized with bleach or another cleaner before and after the experiments. The strain of E. coli being used is HB101 and is a safe strain for humans.
Solutions that have been contaminated with bacteria will be removed and the containers they were in will be sanitized with the 70% ethanol or a 10% bleach solution as well as RNAase. Additionally, participants will wear gloves and safety goggles throughout the experiment/testing and will thoroughly wash their hands before entering and leaving the lab area.
After doing research on the enzyme oleate hydratase, we predict the quality will vary after it gets purified from E. coli.
Supplemental research is suggested to include a more diverse spectrum of enzymes and explore their different characteristics.
Some next steps we are considering are experiments that help determine what environmental conditions could be changed to increase the effectiveness of oleate hydratase. In the future, we could also figure out whether constructing the detergent solely of enzymes would increase the amount of the stain and range of stains that get removed. To make washing off the detergent easier, we could also look at the impact of increasing the solubility of oleic acid.